Eukaryotes. Eukaryotes comprise organisms that differ from prokaryotes in having a compartmentalized cytoplasm with membrane-bounded organelles, including a nucleus. The nuclear envelope separates the two major compartments, nucleoplasm and cytoplasm. Chromosomes carrying the cell's genes in the machinery to express those genes resides inside the nucleus. Most eukaryotic cells have endoplasmic reticulum, the site of protein and phospholipid synthesis. A Golgi apparatus adds sugar to the membrane proteins, lysosomal proteins, and secretory proteins. Lysosomes, compartments containing digestive enzymes, and peroxisomes, containers for enzymes involved in oxidative reactions. Most also have mitochondria that convert energy stored in the chemical bonds of nutrients into ATP. Cilia and flagella are ancient eukaryotic specializations used for motility or sensing the environment. It is important to understand how the eukaryotic cell works, because it allowed the rise of the multicellular organisms. For it, we need to know how every part of the cell works. Plasma membrane. The plasma membrane is formed by a lipid bilayer that is hydrophilic inside and hydrophobic outside. The main molecule in this membrane is a phosphoglyceride, formed by an alcohol and a phosphate, called head, and a glycerol and a fatty acid, called tail. The head is the outside of the lipid bilayer, and the tail inside. In addition, there are some associated proteins that help the plasma membrane do its work, like RAS, SRC peptide, Ty1, annexin, prostaglandin synthase, cadherin, and cadenin. The function of the plasma membrane is control entry and exit of the substances in and out the cell. It's made from three types. Pump are enzymes that use energy from adenosine triphosphate, ATP, light, or rarely other sources to move ions, generally cations, and other solids across membranes at relatively modest rates. Pumps are also called primary active transports because they transduce electromagnetic or chemical energy directly into transmembrane concentrations gradients between membrane-bound compartments. Carriers provide passive pathways for solids to move across membranes down their concentration gradients from a region of higher concentration to one of lower concentration. Each conformational change in a carrier protein translocates a limited number of small molecules across the membrane. Carriers use ion gradients created by pumps as a source of energy to perform a remarkable variety of work. Translocation of anion down its concentration gradient can drive another ion or solute up a concentration gradient, so these are called secondary transports. Channels are ion-specific pores that typically open and close transiently in a regulated manner. Open channels are rightly specific but passive transports, allowing a flow of an ion or other small solutes to pass quickly across the membrane, driven by electrical and concentration gradients. The movement of ions through open channels controls the electrical potential across membranes, so that changes in channel activity produces rapid electrical signals in excitable membranes of nerves, muscles, and other cells. Cytoplasm. The cytoplasm is viewed not as a solution, but as a dynamic gel, a sponge-like network of cross-linked fibrous structural elements, that partitions the intervening liquid into a series of interconnected interstices, channels, or pores. Compartmentation with the cytoplasm includes cytoplasmic microdomains organized by the protein-water network as well as organelles and endomembranes. Cytoskeleton and motility apparatus. A cytoplasmic network of 3 protein polymers, actin filaments, microtubules, and intermediate filaments, maintains the shape of most cells. Each polymer has distinctive properties and dynamics. Actin filaments and microtubules provide tracks for the ATP-powered motor proteins that produce most cellular movements, including locomotion, muscle contraction, transport of organelles through the cytoplasm, mitosis, and the beating of cilia and flagella. The proteins are also used for highly specialized motility processes, such as muscle contraction and sperm motility. Actin filaments and microtubules have much in common, including their evolutionary origins in prokaryotes. Both assemble spontaneously into polymers that are used as tracks by molecular motors. The protein subunits of both polymers bind the nucleoside triphosphate, adenosine triphosphate (ATP) in the case of actin, and guanosine triphosphate (GTP) for tubulin. After polymerization, hydrolysis of these bound nucleotides and dissociation of the gamma phosphate stabilize the polymer, much more so in the case of the microtubules than in the case of the actin filaments. Both polymers can turn over rapidly in cells or remain as stable components. Cells use many proteins to regulate the assembly of these polymers. Some proteins bind to the cytoplasmic pools of the subunit proteins. Others initiate the assembly, some stabilize the polymers, others sever or depolymerize them. Still others link the polymers together or to other cells constituent. During divergent evolution from the common ancestor, contemporary organisms came to use actin filaments and microtubules for some of the same functions. For example, separate the chromosomes in eukaryotes while homologues of actin separate plasmids in bacteria. Actin filaments are tracks for long-distance transport. Actin filaments are tracks for long-distance transport in plants, whereas microtubules serve the same purpose in animal nerve cells. Actin filaments and microtubules cooperate with a third polymer called the intermediate filaments to form a cytoskeleton, which resists deformation and transmits mechanical force. Microtubules are rigid, hollow reinforcing rods that sustain both compression and tension. These mechanical properties make microtubules useful for supporting asymmetrical cellular processes and for bidirectional traffic generated by the motor proteins kinesin and dynein. Actin filaments are more flexible, so they must be cross-linked into bundles to bear compression forces or support asymmetrical process. Right tensile strengths allows actin filaments to bear forces produced by myosins. Intermediate filaments are flexible cables that have the considerable tensile strength but little capacity to resist compression. Both intermediate filaments and actin filaments reinforce woolly tissues by anchoring cadherins, transmembrane proteins used for cell-to-cell adhesion. Intermediate filaments prevent excessive stretching of cells by external forces in multicellular animals. If intermediate filaments are defective, tissues are mechanically fragile. Peroxisomes. Peroxisomes are membrane-bound organelles containing enzymes that participate in oxidative reactions. Like mitochondria, peroxisomal enzymes oxidize fatty acids, but their energy is not used to synthesize ATP. Peroxisomes are particularly abundant in plants. Peroxisomal proteins are synthesized in the cytoplasm and imported into organelles using the same strategy as mitochondria, but with different targeting sequences and transport machinery. Genetic defects in peroxisomal biogenesis cause several forms of mental retardation. Centrioles. Barrel-shaped structures composed of 9 microtubule triplets that organize centrosomes. Centrosomes. Pair of centrioles and surrounding matrix containing proteins including gamma tubulin that nucleate microtubules and serve as the microtubule organizing center in most animal cells. Lysosomes. An impermeable membrane separates degradative enzymes inside the lysosomes from other cellular components. After synthesis by rod endoplasmic reticulum, lysosomal proteins move through the Golgi apparatus where enzymes add the modified sugar phosphorylate mannose. Vesicular transport, guided by phosphomanose receptors, delivers lysosomal proteins to the lumen of lysosomes. Cells ingest microorganisms and other materials in membrane vesicles derived from the plasma membrane. The contents of these endosomes and phagosomes are delivered to lysosomes for degradation by lysosomal enzymes. Deficiencies of lysosomal enzymes cause many several congenital diseases where substrates of the enzyme accumulation quantities that can impair the function of the brain, liver, or other organs. Ribosomes. Complex of ribosomal RNAs with multiple proteins that catalyzes the synthesis of polypeptides. Cilium is an ancient eukaryotic specialization used for motility or sensing the environment. Endoplasmic Reticulum. The endoplasmic reticulum is the largest membrane delineated intracellular compartment within eukaryotic cells, having a surface area up to 30 times that of the plasma membrane. The endoplasmic reticulum performs many essential cellular functions, including protein synthesis and processing, lipid synthesis, compartmentalization of the nucleus, calcium storage and release, detoxification of compounds, and lipid transfer and siling to other organelles. It also has roles in the biogenesis of the Golgi apparatus, peroxisomes, and lipid droplets, and helps mitochondria to divide. Approximately 1/3 of all cellular proteins are imported into the lumen of the endoplasmic reticulum or integrated into endoplasmic reticulum membranes. Import occurs at rates of 2 to 13 million new proteins synthesized per minute. The endoplasmic reticulum retains some of those imported proteins for its own functions. Some are degraded, and others are exported into the secretory pathway for targeting to other compartments within the cell. When the endoplasmic reticulum is associated with ribosomes, it is called the rogue endoplasmic reticulum. When doesn't, it is called the smooth endoplasmic reticulum. Mitochondria. Mitochondria consist of two membrane-bound compartments, one inside the other. The outer membrane surrounds the intermembrane space. The inner membrane surrounds the matrix. Each membrane and compartment has a distinct protein, composition, and functions. Mitochondrial enzymes use most of the energy released from the breakdown of nutrients to synthesize ATP, the common currency for most energy requiring reactions in cells. This efficiency process uses molecular oxygen to complete the oxidation of fats, proteins, and sugars to carbon dioxide and water. A less efficient glycolytic system in the cytoplasm extracts energy from the partial breakdown of glucose to make ATP. Mitochondria cluster sites of ATP utilization, such as membranes engaged in active transport, nerve terminals, and the contractile apparatus of muscle cells. Mitochondria also respond to toxic stimuli from the environment, including drugs used in cancer chemotherapy, by activating controlled cell death called apoptosis. A toxic cocktail of enzymes degrades proteins and nucleic acids as the cell breaks into membrane-bound fragments. Defects in this form of cellular suicide lead to autoimmune disorders, cancer, and some neurodegenerative diseases. Secretory vesicles. Endocrine, exocrine, and neuronal cells use a third sorting pathway from the trans-Golgi network to concentrate and package selected proteins in storage granules before they are discharged from the cell in response to hormonal or neurostimulation. This regulatory secretory pathway stores and discharges on command many proteins needed intermittently, such as polypeptide hormones and digestive enzymes. These secreted proteins apparently lack a universal sorting signal. Instead, secretory granule formation appears to involve physical sorting, selective retention, and condensation of the secretory proteins through charge neutralization, protein aggregation, and active extrusion of ions. Golgi apparatus. The Golgi apparatus performs three primary functions with the secretory membrane system. First, it is a factory to synthesize the carbohydrate chains of glycoproteins, proteoglycans, and polysaccharides secreted by plants in preparation for their biological functions at the cell surface. Second, the Golgi apparatus is a protein sorting station for the delivery to many cellular destinations. This includes transport to the plasma membrane, secretation to the cell exterior, sorting to endosome lysosomal system, or retrieval back to the endoplasmic reticulum. Third, the Golgi apparatus synthesizes sphingomyelin and glycosphingolipids. These lipids associate with cholesterol and influence protein sorted in the Golgi apparatus and plasma membrane. Chloroplast. Photosynthetic bacteria and chloroplasts of algae and plants use chlorophyll to capture the remarkable amount of energy carried by single protons to boost electrons to an excited state. These right energy electrons drive a chemiosmotic cycle to make nicotinamide adenine dinucleotide phosphate, NADPH, and ATP. Photosynthetic organisms use ATP and the reduction power of NADPH to synthesize 3-carbon sugar phosphates from carbon dioxide. Glycolytic reactions running backwards use this 3-carbon sugar phosphate to make 6-carbon sugars and more complex carbohydrates for use as metabolic energy sources and structural components. Some bacteria and archaea, such as Allobacterium allobium, use a completely different light-diving PEMP lacking chlorophyll to generate a proton gradient to synthesize ATP. In that case, retinol associated with bacterial rhodopsin absorbs light through dive proton transport. Plant cell wall. The cell walls of land plants are composite materials consisting of cellulose, other polysaccharides, and glycoproteins. Wood and cotton are two familiar examples of cell wall material that's left behind after plant cells have died. Like the extracellular matrix of animals, plant cell walls not only provide mechanical support but also may influence development. Because of these robust cell walls, plant cells are not motile. Therefore, the morphology of plants is established by the orientation of the cell divisions that occur during their development. Two types of forces act on cell walls. Internally, The vacuole the plant cell applies a radiotruger pressure on the order of 1 atmosphere. Cell wall also resists a variety of external mechanical forces that tend to deform the cell. Nucleus. The nucleus rouses the chromosomes together with the machinery for DNA replication and RNA transcription and processing. Nuclear envelope. The nuclear envelope provides a selective permeability barrier between the nuclear compartments and the cytoplasm and acts as a platform that helps organize the chromosomes in discrete functional domains. The barrier keeps pre-M RNAs in the nucleus until they are fully processed and licensed for export so that only mature mRNAs are delivered to ribosomes in the cytoplasm for translation into protein. It also provides an additional level of genetic protection and control since various chromosomal events, including DNA replication and expression of certain genes, are regulated, at least in part, by changes in the ability of factors to move between the cytoplasm and nucleus. Nuclear pore. In a typical ground cell, nearly all traffic between the nucleus and cytoplasm passes through approximately 3,000 channels, called the nuclear pore complexes that bridge the inner and outer nuclear membranes. Nuclear pore complexes have a scaffold consisting of 3 staked rings each with eightfold symmetry. Cytoplasmic and nuclear rings flank a prominent spoke ring that is intimately associated with the pore membrane linking the inner and outer nuclear membranes. The nuclear ring is anchored to the nuclear lamina. A less prominent fourth luminal ring surrounds the pore membrane in the nuclear envelope lumen. The minimum diameter of the central channel through the pore is approximately 40 nanometers, and the channel is approximately 50 to 70 nanometers long. Nucleoplasm. The cellular region within the nuclear envelope. Nucleolus is the most conspicuous and best characterized nuclear subdomain. Most mammalian cells have no to 5 nucleoli, with your specialized region 0.5 to 5 micrometers in diameter surrounding transcriptionally active ribosomal RNA gene clusters. Nucleoli are the sites of most steps in ribosome biogenesis, from the transcription and processing of rRNA to the initial assembly of ribosomal subunits. The ribosome is a complex macromolecular machine with 4 different structural rRNA molecules and approximately 85 proteins that are assembled into 2 subunits. Transcription of rRNA by RNA polymerase I comprises nearly half of total cellular RNA synthesis in some cell types. This wide level of synthesis is necessary to produce approximately 5 million ribosomes in each cell cycle, more than 30 every second in budding yeast.